A warm welcome to all of you. Like in all animals, plants are also attacked by pathogens, which impair their growth, development, reproduction, and in some cases may cause death. Hence, to protect themselves, plants also have their own immune system. The immune system in plant is what I'm going to explain in this presentation. Now first, let us see the different life strategies the pathogen uses to infect the plant. The bacterial pathogen enters the plant body through stomatas or through hadathodes or through lenticels or through wounds caused by mechanical effect or by human beings or through frost, etc. And once they enter into the plant body, they multiply in the intercellular spaces using the nutrient of the plant and cause infection. The nematodes and aphids, they insert their stylet into the plant cell, suck the nutrient and cause deformity in their morphological structures and thus impair the growth and development of the plant. The fungi can directly enter the plant epidermal cells or extend their hyphae on the plant surface, in between the plant cells, or through the plant cells and cause infection. The surface growing pathogenic, symbiotic and oomycetes invaginate haustoria into the host cells and cause infection. To protect themselves from these pathogens, the plants have their own immune mechanism. One should remember plants do not have mobile defender cells and a somatic adaptive immune system which is commonly seen in animals. Instead, they depend on the innate immunity of the cell and on the systemic signals originating from infection sites. It is now clear that there are two methods of plant immune system. In the first case, the plant uses transmembrane pattern recognizing receptors that respond to slowly evolving microbial associated molecular patterns. And in the second method, which largely acts inside the cell using the polymorphic nucleotide binding and leucine-rich lipids proteins encoded by the specific resistant gene. One should remember this type of disease resistance, that is NBLRR mediated disease resistance, is effective only on pathogens that can grow on living cells and not on the necrotrophic pathogens that kill the host tissue during infection. The whole mechanism of plant immune system can be explained by this zig-zag model of plant immune system. There are four phases in plant immune system. In the first phase, when any pathogen invades a plant, pathogen-associated molecular patterns are released. Now, these pathogen-associated molecular patterns or microbial-associated molecular patterns, these are recognized by the pathogen recognizing receptors or co-pathogenic recognizing receptors present in the transmembrane of the cell. This causes the production of the reactive oxygen species and antimicrobial compounds which combat with these pathogens and destroy them and stops the infection. So thus it causes the pathogen triggered immunity, that is PTI. So in the first phase, the PAMs are released by the pathogens. The pattern recognizing receptors of the plant identify these PAMs, destroy them, and induce the PAM triggered immunity. Now, a successful pathogen, now what it does is it releases these effectors. Now, these effectors react with the PTI mechanism and stops it. So the defense mechanism of the plant is checked now. And this causes the effector triggered susceptibility in the plant and the infection of the pathogen progresses. So in the second phase, the successful pathogen deploy effectors. Effectors can interfere with the PAM triggered immunity and induce effector triggered susceptibility, and hence the infection proceeds. Now, a plant having a resistant gene or a resistant variety plant, it has got these genes which can interfere with these effectors. So such a resistant plant having the resistant gene for that particular infection releases the intracellular NLRs or NBLRR, that is nucleotide binding leucine rich lipid proteins. Now these NBLLR, they attack the effectors, form a complex, and the NBLLR becomes activated, and then this causes the effector triggered immunity. This effector triggered immunity is nothing but an accelerated or amplified PAM triggered immunity, wherein again all these the molecular patterns released by the pathogens are destroyed by the plant immune system. So this is how the infection stops. Now this is the second mechanism, that is the second method where it is occurring inside the cell, and this type of ETI is seen only in those plants which is having the resistant gene for that particular disease. We'll just recall the phase 3. In the phase 3, the resistant plant releases the nucleotide binding leucine rich lipid proteins. The nucleotide binding leucine rich proteins bind with specific effectors and become activated, and this induces the effector triggered immunity, which is nothing but the accelerated and amplified PAM triggered immunity resulting in disease resistance. Now, effective pathogen, what it does is it wants to survive. So it will again modify its effectors. So the natural selection drives the pathogen to avoid the ETI either by shedding or diversifying the recognized receptors gene, or by acquiring additional effectors that suppress ETI. And natural selection results in new resistant specificity so that ETI can be triggered again. So all these four phases can be illustrated by this zigzag model of plant immune system. Now, when the infection occurs in the first stage, the PAMs are released. Now, these PAMs are recognized by the pattern recognizing receptors. So, the defense mechanism of the plant increases and the infection stops. Now, to counteract this PTI, that is PAM triggered immunity, the pathogen releases effectors. Now, these effectors will nullify the PTI, so the infection proceeds. Now, to counteract these effectors, the resistant gene of the plant releases the NBLRR. These NBLRR combine with the effectors and results in effector triggered immunity, which is nothing but amplified PAM triggered immunity, and the infection stops, and hence the defense mechanism increases. Now, the natural selection drives the pathogen to modify these effectors. So you can see here some of the effectors are shown blue in color. I mean, these are the modified effectors. So these modified effectors can override this ETI, and again the infection increases. So it becomes effector triggered susceptibility, and then again the plant will modify its resistant gene so that it can counteract these new forms of effectors, and then the defense mechanism increases. So thus, this fight between the pathogen and the host plant is always going on. Plant wants to protect itself by its immune system, and the pathogen wants to invade the plant for its survival. So this fight is going on, and it goes on, and when the plant is successful, the plant is resistant to the gene. When the pathogen is successful, it causes disease to the plant. So this is how the immune system works in plants. Thank you.